guys, welcome to the classroom. For those of you who have been learning remotely, I got a webcam, so I'm going to see how this works because we are actually starting to get into the things that you probably came to genetics for in the first place, and that is to learn about Punnett squares. Who likes Punnett squares? Okay, so I'm starting off with simple patterns of inheritance, and pardon me while I get used to this. Looks like I got to adjust the camera just a little bit. There we go. Alrighty. So let's see. Pangenesis. This is the idea that seeds are produced by all parts of the body and are then transferred back to the offspring. Probably never heard of these people before. Joseph Kohlreder. He completed the first studies of genetic crosses with tobacco plants. And I talked a little bit about tobacco plants when we were doing the disease bacteria study. He concluded that offspring are an intermediate of their parents. And this is consistent with the theory of blending inheritance. And again, this is probably stuff that you have not heard of before, but don't worry, we'll get through it. This guy you have heard of, Lamarck. He hypothesized that species change over the many generations by adapting to new environments. And he said it was more behavioral. These behavioral changes are going to modify traits that can be passed on. And we always use the example with the giraffes here. Way back in the day, we did have giraffes that had long necks, and we had giraffes that had shorter necks. Now, the short-necked giraffes, as you can see in that picture, they can't reach the leaves on all the trees. So they are kind of, at the mercy of hoping that these taller giraffes will either give them some leaves, or maybe the taller giraffes will just eat the leaves at the top of the tree, and maybe they'll be able to get food. Over time, those long-necked giraffes, they weren't like people. They didn't help everybody out. So they ate what they wanted, and the little short-necked giraffes ended up starving. That is not a giraffe child. The parents would have fed it. That is an actual grown adult giraffe. So now we only have long-necked giraffes. But Lamarck believed that maybe this short-necked giraffe would stretch his neck. And as he was stretching his neck, that trait would then be passed on to the offspring. And we found that that's not how it works. Let's say you're a bodybuilder and you go to the gym every day of the week, sometimes twice a day for 10 hours every day. Maybe you're like The Rock. The Rock's children are not going to be bodybuilders unless they go to the gym and work out as well. You can't pass on that degree of muscle in your offspring. So behavioral changes do not get passed on. It is only things that are actually in your genes. Particulate inheritance is the idea that determinants of hereditary traits are transmitted intact from one generation to another. Gregor Mendel, you've heard of him, he's the father of genetics. He used statistical analysis to carefully design breeding experiments to arrive at the concept of a gene, and we're going to study his work in this section. You've definitely learned about Gregor Mendel. I teach about him in regular biology. Some basic terms just to review, because again, you guys have heard this all before. Traits or characters, these are features of an organism. Self-fertilization, this is what Mendel did for a little while. It's a plant can pollinate itself. True breeding exhibits the same trait after several generations, and cross-fertilization is one plant will fertilize another plant. The P generation stands for the parent generation. F1 generation, a lot of times we say it stands for first generation. The F actually stands for filao, kind of a weird term. I believe it's Latin. Everything is Latin in science, right? Monohybrids, these are single traits that are crossed. A dominant is a trait that's displayed. Recessive is going to be a masked trait, unless, of course, you have two recessive traits. Genotype is a genetic composition of an individual. Heterozygous is two different alleles. Homozygous is two of the same alleles. And phenotype are characteristics of an organism. These are things that you can observe. Mendel's laws and the chromosome theory of inheritance. He talked about hybridization. This is where two individuals with different characteristics are mated or crossed. So, for example, a purple plant crossed with a white plant. And then he carefully analyzed the numbers of offspring carrying specific traits, leading to the formalization of two principles. The first one was the law of segregation, and that states you have two copies of a gene. They segregate from each other during gamete formation, which happens in meiosis. Gametes are the sperm and egg cells, and they go from parent to offspring. Mendel observed approximately a 3-to-1 phenotypic ratio between dominant and recessive traits. So if you have a regular Punnett square, 3 out of the 4 times you're going to have that dominant trait, and then a quarter of the times you're going to have that recessive trait pop up. So we're going to cross a true breeding tall plant with a true breeding dwarf plant, where tall is going to be dominant. Then we're going to cross the F1 generation through self-fertilization to determine the F2, and we're going to look at the genotypic and phenotypic ratios. So we're going to hop right back into Punnett squares. So true breeding means that they are going to be homozygous. So for my parents here, I have a true breeding tall plant. I'm going to use capital T's. Moment you've all been waiting for, right? And a dwarf plant. Dwarf is obviously just another term for short plants. So I have 2 capital T's and I got 2 lowercase t's. And you know you want to have 2 letters in each box. Preferably you are writing the capital letter if there is one first. So I push those capital T's down, I push the lowercase t's over. And that is what I get in my F1 generation. So when I cross my F1 now, it doesn't matter which 2 boxes I pick because they all have the exact same genotypes. So I have a capital T and a lowercase t, and a capital T and a lowercase t. So self-fertilization here, what are we going to get for our F2? Genotypic ratio. So for genotype, I'm going to have a 1 to 2 to 1. You always start with your homozygous dominant, and I have one of those. Then you're going to go with your heterozygous next, and I have 2 of those. And I have 1 homozygous recessive. Now, phenotypic ratio, this is what Mendel was talking about when he said he came with a 3-to-1 ratio. For the phenotype, that's what these things are going to end up looking like. Are they going to be tall or are they going to be short? Anytime you have a capital T, that's going to represent a tall plant. So I have a capital T here, here, and here. So I have 3 that are going to be tall, and I have 1 that has no capital T's, so I have a 3-to-1 ratio there. You always do dominant traits first and recessive traits second. That was just a blank slide in case I needed it, but I didn't. So determining genotype, you can do a test cross. A test cross is used to determine the genotype of an unknown cross with the homozygous recessive trait. You always want to use homozygous recessive because if there is a dominant allele there, it's going to show up, and that will definitely tell you what that unknown genotype is. If you use a heterozygous trait here, you're not going to know what the genotype is. Analyzing 2 traits, this is a 2-factor cross. You're going to be examining 2 different traits simultaneously. That's a dihybrid cross. It's just another name for it. If you took AP Bio last year, or if you were in honors bio, a lot of times you'll do dihybrids at the very end. In most academic bios and in general bio, I don't go over dihybrids because it's not keystone tested, and it takes some time to just do regular Punnett squares, monohybrid crosses. So inheritance patterns. This is where 2 genes are linked in some way so that the variants that occur together in the parents are always inherited as a unit. And the 2 genes are independent of one another so that their alleles are randomly distributed into the gametes. So this is what a dihybrid cross looks like. Here we're looking at color and we're looking at the seed texture. So is it round versus smooth, or is it green versus yellow? So if I were to guess here, I would imagine that the capital Y is going to be yellow, capital R is going to be round. So you would have one for each. So you'd have a capital Y with a capital Y with a little r, little y with a big R, little y with a little r. And you end up with actually 6 squares. And we're going to go through some dihybrid crosses later on. I'm going to do monohybrids for a while first, but this just gives you an idea of what you'll be doing. Yeah, you'll have these 16 block Punnett squares to work with. Law of independent assortment. This is where alleles of different genes assort independently of each other during gamete formation. And you can only see this in dihybrid crosses. In monohybrid crosses, you don't notice it. The chromosome theory of inheritance. It states that chromosomes contain genetic material passed on from parent to offspring. Chromosomes are replicated and passed on from parent to offspring. The nucleus of a diploid cell contains 2 sets of chromosomes, one maternal and one paternal chromosome. At meiosis 1, a member of each chromosome pair segregates into the other daughter nucleus, and each is going to result in a haploid cell. Gametes are haploid cells that combine to form a diploid cell during fertilization. Again, your gamete cells are the sperm and the egg. They have to be haploid because if they were diploid cells, you would have double the amount of chromosomes, would not result in a baby. It would be too many. It would be miscarried. Probability. You'll be determining probability here in a little while. It's the chance that an event will have a particular outcome. So it is the number of times an event occurred over the total number of possible outcomes. So the number of times an event occurred, this is the one you were studying, and this is how many total you did. Total experiments. Pedigree analysis. Remember doing pedigrees? Maybe, maybe not. This is analyzing inherited traits over generations. If you're going into something like genetic counseling, you'll be doing a lot of pedigree analysis. You can track not only normal traits like your eye color, but you can also track diseases. And a lot of times if you're looking at genetic counseling, that is what you're doing. You're figuring out if it is a genetic condition, where did it originate from, and then you'll calculate the odds for the parents so they know if they should attempt to have a child, what the possibility is for their child to have that certain disease. So the first generation up here, we have a man. Squares are going to be men, circles are going to be females. So we have an affected man here, and I'll just put a little A up here so that you know he is affected. Anytime something is shaded in, they are going to have that specific disorder. They went on to have 4 kids down here. They had a female who had it, 2 boys who did not have it, and they had a male that had it. Over here we have another set of parents. The male did not have it, but the female did. She was affected. They had 2 children. Both of them were girls. One girl did have it. One girl did not have it. So whenever we move on down to look at their kids, they also had 2 girls, one of whom had it and one of whom didn't have it. And this is a really interesting pedigree here because you can see there's 2 affected parents, but whenever we come down to the bottom here, one did have it and one didn't have it. So now you're looking, was this a dominant trait or is it a recessive trait? Take a minute and think about that. If you have 2 affected individuals, how on earth could they get a child who does not have it? What would their genotypes have to be? Going back to the beginning here, talking about dominant and recessive traits, if you have a dominant trait, you only need 1 allele to have it. If it's a recessive trait, you have to have 2 alleles. So, in order for them to have a child who does not have this condition, this particular disorder has to be a dominant disorder. So that means that these 2 parents here were going to be heterozygous. So their second letter there would have been a little a. Coming down to this daughter here, we don't know if she has 2 dominant alleles or if she has a dominant and a recessive, and that doesn't really matter. All we know is that she does have it, so she has at least 1 capital A. But this daughter that doesn't have it, is going to be two little a's. So this is going to be an autosomal disorder. It does not affect one gender over the other, but we know that everybody who is clear or not filled in is going to be a homozygous recessive person. So that means that all these parents, in order for them to have kids that didn't have it, they were also heterozygous. If the parent was homozygous dominant, every single one of these kids down here would have also been affected with the disorder. Now looking at these kids here, we know that this mother is homozygous recessive, so that means that this daughter has to be heterozygous. So she has a chance of having a kid with it, or maybe not. It depends on what her husband is. So like I said, shaded symbols are going to be infected with the disorder. Clear symbols are not infected. If you have a half-shaded person, this is a carrier, but this symbol is not always used. So half-shaded would just be like this, and it just tells you if they are heterozygous or not, is basically all it's doing. So we'd say that that's a female, and automatically we could tell you that her genotype is heterozygous. And most of the time, you're going to have half-shaded people if the disorder is a recessive disorder. And that is allowing you to know that while she doesn't actually exhibit that trait outwardly, she could have a child that has that trait. Most human genes are located on chromosomes called autosomes, and when we went back to that other pedigree, I said it's an autosomal disorder, which means that both men and women can inherit these the same way. You also have sex-linked disorders, which we'll get into those as well. Other genes are located on the sex chromosomes, and that ratio is going to differ in males and females. Most of the time, if it is a sex-linked disorder, it is going to affect the boys more often than it will the women. In mammals, males have an X and a Y. Females are going to have two X's. Many sex differences are due to the presence of those sex chromosomes. The SRY gene is located on the Y chromosome, plays a key role in the developmental pathway that leads to maleness. I don't know if that's really a word or not, but that is what makes guys a guy instead of a girl. So that little Y chromosome, even though it is extremely tiny, it has a lot of genes on it that play a big role in making boys boys. Insects, they have an XX and an XO system. So whenever we start doing sex-linked disorders or sex-linked Punnett squares, I'm going to tell you if it is a human or if it is a chicken or a fly, because the X's and the O's or the X's and the Y's, they're all going to be different depending on the species. So females are going to be diploid and the males are going to be haploid. Look at the size difference between that female and the male. I'm not saying that females are more important, but in the insect world, they are more important. Females are the ones who have the babies and they have the whole population there. They kind of, think about queen bees, they run the show and the boys are just the workers. Birds and some fish have a ZW system, where females are going to be ZW, males are going to be ZZ. So it's a little different than people. In some reptiles and fish, sex is determined by environmental temperatures. For example, alligator eggs incubated at 91 degrees Fahrenheit, they produce males, while at a temperature below this, you'll get females. And if it's above that 91 degrees, most of them are going to be females. You may have a male in there every now and again, but he has to be right at 91 degrees in order to get the boys. In the animal world, you're trying to have more females so that there's a better chance that you can pass on those traits and those have more babies. If you have more males, males can't have kids, so you're kind of stuck. X-linked traits. Males are more likely to receive a recessive X-linked trait than a female due to the fact that females have two X's. So if we're talking specifically about humans here, if you have one X that has a recessive trait on it and you're a girl, you still have another X, which gives you the possibility of being a carrier for the disorder, but not actually exhibiting the disorder. Whereas boys, if the mother has an X that is recessive, they're going to get whatever disorder that is. Like color blindness is a huge one. It's an X-linked recessive disorder, and that is why 9 times out of 10, boys are going to be more prone to being colorblind than girls are. Not saying that you can't have a girl that's colorblind, it's just less likely to happen. Hemizygous is a term we use to describe males having a single copy of an X-linked gene. So all the boys are hemizygous. Different types of inheritance patterns we can have. We have incomplete dominance, which is a cross or blending on the traits. Codominance expresses both alleles simultaneously. They are both dominant. You don't have one that's recessive. And sex influence is going to show a different phenotype depending on the sex. Gene mutations are going to cause many inherited diseases to have philotropic effects, which means that a mutation on a single gene is going to have multiple effects on individuals' phenotype. An alpha-1 deficiency. This is an inability to prevent the activity of protease enzymes, which causes liver damage and emphysema, which is a lung disorder, and it is a recessive disorder. This is a fun word to say. Phenylketonuria, perhaps? It's the inability to metabolize phenylalanine, which causes severe intellectual disabilities and physical degeneration. If caught early enough, it can be prevented by following a phenylalanine, which is a plant-free diet. How often are you ever told, don't eat plants? I don't think ever. In fact, it looks like they're trying to move more towards us eating plants, because when I go shopping at Walmart, I see there are plant-based food. You know, you have those hamburgers at Burger King now, which is all plant-based, and it's supposed to taste just like a regular hamburger. And the crazy thing is that most of the time it does. Texture-wise, it's slightly off, but taste-wise, it's pretty on point. Tay-Sachs disease, you've heard of that one before. It's a defect in lipid metabolism, and it's going to lead to paralysis, blindness, and early death. So one little trait being off, this is what we're talking about, how it can affect multiple things. Another recessive disease. And that is it as far as our simple inheritance goes. So next time we'll be getting back together. I'm going to do some examples going through just monohybrid crosses and in general how to do Punnett squares. All right, catch you later.